How's everyone? Morning. So, how's everyone today? Okay, yeah, Pakhi, hello. So Pakhi, would you like to give an update?
Yeah, hi,
Hi.
hello. Am I audible?
Yeah.
Yeah, so over the last week, last week, I had given my update where I was generating the nodes for the graph. So then I was also able to incorporate the position, like the coordinate position of the different cells. There was one another CSV file, and then now I'm using the Euclidean distance between two nodes to generate like the weight for the edge between those two nodes.
All right.
Uh, yeah. So I have also set like a threshold for that. Like if the cells are too far away, they will not necessarily have an edge between them because then like that's not required. So I mean, this is like a rough idea for now. Like when I actually feed it into the data model, when I feed it as a data set, then I will see if I need to like adjust some parameters over there. And then in the NDP network, I had told you like a couple of meetings ago, I was facing some issues, errors in running. I was able to fix them as well, and now like the model is training, but it is taking forever to train. By the way, I was
Oh,
able to train it like a few days ago, and now I started it like an hour ago. It still hasn't trained. So yeah, I will see how that goes.
all right.
Uh, yeah, and yeah, that was about my update. So I'm just right now working on like the NDP code. So I was able to like finish the function for the multi-layer perceptron, and now I'm just like working on the training.py file, like which will actually train the model.
All right. Yeah, I think that's. So, how are you running? How are you training the model? The NDP model. How is that?
Okay, so they had provided some code like for training, but like we don't need all of that. So like I just took the necessary functions from it, and then I modified them a little. So 1 second, I will actually share my screen.
Okay. Okay.
Is it visible?
Yes.
Yeah. So here, like they have a bunch of functions in the NDP code itself. So out of these, we didn't need all of them. So I just took the necessary ones, and like I've changed the code also in a few functions. I have pushed this code to a repository, not the latest changes, but like the this NDP file is there in this, and in the train.py file. So they're using this function called fitness, where they're basically fitting the model with the right parameters. That's why the name. And yeah, so this function is basically for the growing graph. This is for that, and this is for fitting that graph with the multi-layer perceptron, which was we were using for determining where the nodes will be added and the weights of the edges, and also this is that function. And this is another function that I was like experimenting with. So I will give updates about this. This is also for they we are basically using three MLPs. One MLP will tell where the node will be added. Other one will tell what will be like the weight of the edge, because we have like a weighted graph here, and it is weighted and directed as well. So this will manage all that. So I'm still working on this one as of now.
Okay. Looks like they have an interesting comment in here under worm in the worm growth function. High fitness value.
Sorry.
Yeah. Go back to the where you
Yeah.
were. Yeah.
Yeah.
So yeah, it looks like they have an interesting comment in the worm growth function. High fitness value means the graph has small worldedness. Returns a scalar measure of small worldness of a graph G. Omega ought to be close to 0 and or sigma is greater than 1. So this is where you're basically getting a measure of what they call small worldness, which is where you have a lot of centralized hubs. You have a very small diameter of the graph. So if you go from one end of the graph to the other, it's a small number of hops. And so what you're basically, well, what that means is that if the graph is more fit, you should sort of get I guess, a small world graph, which I guess, make kind of makes sense. That, the graph gets consolidated and the distance between, like the path length, that they call, which is if you go from one end of the graph to the other, that, and you just make random hops across it, that's the path length. That path length then is minimized, and then you get the small world uh, architecture, which has a lot of these hubs, a lot of these centralized nodes. It's not, it doesn't necessarily follow that, the best graph is small world, or, certainly an optimized graph can be small world, but you can optimize it for other types of topologies. And that's maybe kind of where, you know, what they're doing here is maybe, I don't know if it's as biological as it could be. Let's just put it that way. So you could have, like, you could make the fitness, or you could make it so that the optimized graph is actually what they call scale free, which means that there are many different, uh, type numbers of connections to each node. So yeah, it's definitely something that I wouldn't worry about now, but that would be interesting to explore with the code. And then these are, of course, the sigma and omega values, which I guess are related to fitness. So yeah, this is interesting. I'm glad you're getting into the code and kind of looking at how to run it and train it. So you actually train on a data set, or do you train on, like, what is it that they have that they're actually training on? Is it just kind of like
No,
idealized
no. So
graphs?
like in their repository, they have like a NumPy array for like they experimented with different kind of models. Like they have an XOR gate and then a loner model, all those things. So for that, they have like a NumPy array. I like it is over here. Yeah. So they're using like these arrays for that. But like in our model, I was thinking of like that they had shown. 1 second. Like yeah, so I'm planning on passing these nodes along with their like weighted edges. So for the edges also, I was planning on like having vector embeddings at each node, which will have like the lineage name in the form of a string, and like then along with the coordinates. So like we can get both the edge weights and like which node it should connect to based on the naming of the node. So
Okay,
I'm planning on using this as the data set to train the model.
that's good. Yeah, that's very good. I think that's having that kind of abstraction and then training it on those weights, that should help with C. elegans, since we're kind of focusing on C. elegans. And we can actually get these kind of graphs for other embryos, but then having that framework, we can then like plug those matrices in and use them as, ways to train the model for different types of embryogenesis. But yeah, this is good. This is good.
Thank you.
Yeah. Was there something else you wanted to show us, or?
No, no, this is about it that I've done in the last week.
Oh, that's
I was
good.
busy with a lot of evaluations, so wasn't really able to put much time into it. But like I could get somewhere. Like
Yeah, yeah. So, what are your plans for this coming week?
so for this coming week, I am planning on like finishing up the function for like the training the model and like getting it running, and then once it's trained, then only I will like know how to like adjust the parameters and all according to our like for a more optimized model. So I want to like get it done by this week.
Okay, yeah, that would be good.
Yeah.
Yeah, this is good stuff. Yeah, I'm glad you're able to get this working, and we can dig into it a little bit.
Thank you.
Yeah. So any comments or questions from Hussein or Jesse? Okay. Jesse pulled up a link here, Small World Network. This or more along the lines of what we talked about some time ago in the cybernetics group. So this is actually, it would be this Wikipedia article. Let me see. So it would be this Wikipedia article where they talk about small world networks. This is a kind of an example of what they're getting at here. So a small world network is a graph characterized by a high clustering coefficient. So they use, in graph theory and in network science, they use these parameters to characterize a graph. Usually they'll characterize it by a couple of parameters such as degree, clustering coefficient, and other types of distance metrics. And then, you can actually measure out sort of some of the properties of these topologies. So they kind of look kind of strange because everything's connected to everything else. But the fact of the matter is you can actually characterize these with different types of statistics. And so path length is one. So you might say, if I want to go from this point up here at the top to this point down here at the bottom left that are kind of encircled in purple, you might say, okay, what is my least path length, or what's the shortest path I can take from one of each of these two nodes? And so, the shortest path length here appears to be 1 because you have a connection here. You have many ways to make 2 connections between these 2 nodes, and some ways to make 3 and 4 hops to this node. So you can have like a minimal distance, which is 1. You can have like a mean distance, which is, in this graph, maybe 2.5 or maybe 2, which means that on average, if you take all of the paths possible, you get about 2 hops per, graph. And then you have, 3 hops and 4 hops. So you can actually take, I think here, you can take 5 hops around the edge. But, that's not something, that's an outlier, but that's added into that average. And then you can also optimize the graph for the longest path length as well. And sometimes that's important because if you have a growing network, these edges might disappear over time, or they might reconfigure themselves over time. So, there are a lot of ways to measure path length, a lot of ways to measure centrality, meaning if you take a single node, how central is it to the graph? So how many other things is it connected to? And so if you take these kind of measures, and clustering coefficient is one of those where you basically say, how much is sort of concentrated on this node. If I got rid of this node, how many things would be affected? And so that's going to be, for this node, it's at least 6 nodes are going to be affected. So that's what we might call a hub or highly connected node. And so that's where we come to small world networks, because small world networks have these hubs. They have these hubs of large effect, meaning that they're connected to a lot of other things. And as a consequence, they affect a lot of other things when they change. And so we can count this out. We can get these statistics and describe the topology of these networks. We can have random graphs like this where we have no hubs at all. So in this case, we have things that are connected maybe at most by 4 things. This node is kind of maybe hub-like, but not really. But it's connected randomly, so we don't expect too much structure. And so, there are a lot of ways we can characterize these graphs. And so small world networks is one way to describe this kind of graph. These were discovered in social networks. So, Watson-Strogatz in 1998, they kind of described, or they kind of coined the term small world network. They coined this from aspects of the clustering coefficient, this node-to-node distance, or the shortest path length. And, and so they were able to characterize this in a social network. or like I guess it was an instance of the internet, in fact. But there are all sorts of things having to do with the network structure, and small world networks have a certain aspect to them where they have certain types of cliques that are local, highly connected groups, and other types of subnetworks. And so, we have things like small world networks, we have scale free networks, which I mentioned, we have random networks, and we have other kinds of networks where you can, where the community structure varies and the subgraphs vary in ways that are interesting. So, this is relevant to C. elegans connectome because C. elegans connectome is considered a small world network. There have been papers written on what they call the rich club of neurons in the connectome, and those are neurons that are connected to a lot of other neurons, and so those neurons are key in sort of the operation of some of the functional aspects of the connectome. So it could be like some motor pathway or some sensory pathway, and there's certain neurons that sit at those junctions, and it governs a lot of functions. So that's it's not really related to cybernetics so much. There's some interesting connections between like what we've been doing in network theory or network science for like 30 years and cybernetics, and that might be an interesting foray because you look at these structures and you say, oh, that kind of looks cybernetic like, and it could be, but no one's really ever explored that. So that's the answer to that question. And then Jesse says, I can't remember the guy's name where small worldedness was associated with computation and sort of a finite or limited possibility space. Yeah, I don't know. I'll have to look that up, but I think that's an interesting thread as well. Yeah, it's really about, like these networks, they give you this possibility space. When you have a randomly connected graph, there are a lot of different combinations or a lot of different pathways. And when you have these kind of networks that are selected for maybe in a small world direction or some other direction, like a regularized network, that closes down the number of possible pathways, but sometimes it opens them up. It really depends on, what part of the graph you're in and where you want to go and so there are a lot of things you can do with networks that are really interesting in that regard. So thank you, Pakhi, for your update. And yeah, so Hussein, hello.
Hey,
Hi.
hey. Yeah. So yeah, thanks for the meeting. Thanks. So I mean, I don't have anything to say regarding DevoWorm updates, but I could talk a bit for the breast cancer CT project. I think Dick might be online right now. I should just message him.
He was here earlier.
Wait, is he? Oh, he's not in the room. He's not in the room right now.
No, not
I
right.
See that. Okay. Oh, so he was here. He was
Yeah,
here. Okay.
he was here.
Okay, cool. Yeah, thanks so much. Thank you for the discussions. Small worldedness, and it's like the small world network. I think there was a there was one paper I came across. I think it was okay. I mean, speaking of like the excuse me, okay. One thing I wanted to quickly add was basically on the, on the idea of groups and networks, on how small or how large they are. Basically, exploring the properties of whether they, in what ways they can, when they expand from small to larger ones, how likely are they to share edges, and in what ways and how likely and how highly connected are they? How highly connected are they? Would they share loops?
Yeah.
Whereas there are theories being put theorems and proofs being put forward that in small highly connected networks there have to be loops. There must be a loop, and you being able to define that in all circumstances basically would reduce being able to prove that and being able to show that would not only just be able to make a lot of proofs in theory work in mathematics and logic a lot simpler, but prove very fundamental for a lot of structured tools from a computing and quantum computing point of view. So it's all very, very interesting.
Yeah. Yeah, of course, we know in small world networks, you might know it as sort of the 6 degrees of Kevin Bacon, where it's you have like a bunch of actors and that, or people who all kind of know Kevin Bacon through 6 associations at maximum. So that speaks to these shortcuts that go across the network. You don't have to rely on, and this was something actually done by Stanley Milgram back in, I think, the 60s. He didn't call it Small World Networks, but he sent a bunch of letters out to people in the mail, and he said, forward this to people who you know, and try to get to this, if you think you can get to this destination back in Boston, try to send it to the person who you know and see if we can get the letter back. And they just looked at the number of postmarks on each letter that ended up back at the destination, and they ended up figuring out that it took sort of on average 6 hops. It could take more, it could take less, but that suggests that people's social connections, that relay was around 6 stops on average, which is actually for just sending out letters at random actually quite impressive. You distribute things out on the nodes and you say, how long does it take to come back to the central node? And it doesn't take as long as you might think, even for a network of millions and millions of nodes. So that's kind of the power of small world networks. Now, there are other types of network topologies that are interesting as well, and that's something we can talk about later. But
yeah, absolutely. Okay, thanks. Thank you.
Yeah.
So Dick and I have been looking into the low-dose CT Grand Challenge. Okay, so excuse me, just taking a step back. Our algorithm, our BRCT, excuse me, our algorithms, multiplicative algebraic reconstruction, 2 different versions of them, being used in optimizing the efficiency and effectiveness of the breast cancer detection screening techniques and protocols, as we use the steerable J-sphere technology between X-rays and polycapillary optics. We want to see how effective our algorithms and our approaches are, and in particular, we've been comparing them to the standards of what we found as the low-dose CT challenge. Let me just share my screen.
All right.
Show this. The low-dose CT challenge. There we go. Like Dick was asking me, are we lowest dose?
Yeah.
Getting the writing together on this. So basically, the low-dose CT Grand Challenge would be used to see the diagnostic performance of the different denoising and reconstruction techniques for low-dose patient CT datasets. And this was several years ago, back in 2016, I think. We just wanted to compare our results and our data against what was shown there to see how well we're doing in comparison to the others. So that's very, very, very important.
Right. So this is, could you bring that back up?
No problem.
Yeah. So I just wanted to look through some of this.
Yeah, no problem.
Yeah, so you have patient data, which is sort of what you're training the models on. And then you have contrast-enhanced abdominal CT examinations. All data were obtained on similar models. And then these data were acquired using a flying focal spot. That's just basically describing the training data, and then go down a little bit. I mean, this is just kind of describing how this was done, not necessarily you can do this with other kind of methods. But then you're inserting noise to reduce dose levels. So you're kind of, the dosage is like giving a signal that's higher, and then the noise is taking that away, correct?
Yeah, absolutely. Like, so where does it talk about that? Yeah, denoising or iterative reconstruction in particular. Okay, so basically, when forming the image upon the screen, we want to get rid of like aberrations, blur, other parts of what could be produced that are detracting, deteriorating from what we need to be looking for as we perform the screening processes and procedures. So denoising, or excuse me, denoising is done alongside the deconvolution. So basically, for any sort of imaging, like I think, in particular for screening for breast cancer, like for fMRI, CT scans, but for any sort of image processing and image production, typically the set of steps would be like calibration, then removing gradients, deconvolution, and depending on what your workflow is, denoising would be somewhere there too. So we just wanted to compare our techniques and our approaches to what had already been done from this, because this is long gone.
Yeah.
When was this?
2016, I think.
I think it was 2016. Okay.
2017, yeah. Oh, they have it right out there. The Mayo Grand Challenge, yeah. Yeah, 2016.
So many things. Too many things to keep up with.
Yeah.
Yeah.
Looks like they published it, though, in 2017, so they actually have a paper associated with this. So, yeah, who won the Grand Challenge, I wonder?
I think there was a Kaggle dataset for it. There's something out there. Maybe they put it on Kaggle. Yeah, they did. They did put it on Kaggle.
Okay, good.
Cool. Trying to get your rights. Cool. Yeah. Exciting stuff. Happy to keep you guys posted on all this. Yeah.
Yeah, great. Yeah, that looks really interesting. So on Kaggle, you have you can download. I guess this is the training data you can download, or is this the?
Yeah. The training data can be downloaded, and yeah.
Okay.
Excuse me. Yeah, it can be downloaded. It can all be tested once. So.
Yeah, it looks great. Thank you.
Okay. Thanks.
All right, so that looks good. And so we have, I don't know if we have, okay, Pakhi had to leave. Thank you, Pakhi.
Yeah. In other news, like the work with Alter Learning is going very well. We're being very thorough with plans and designs and initiatives. So I'm the AI team leads, and goodness, I wish like this is for the company, for Alter Learning, for the ed tech startup that I'm working with. So things are going well with that. So
well, that's good. Morgan isn't here today, but I'm going to do something that he's interested in. He's talked about this a number of times in our Saturday meetings and in the Slack. This is the Build a Cell, project. I'm not really sure what, they have a lot of things. They have a seminar series, they have workshops, they have a set of working groups. And what they're doing is this open collaboration supporting the science and engineering of building synthetic cells. So this is all synthetic biology type stuff. This is where they're trying to build cells, and you can see that they're using their fluorescent proteins to sort of verify their cells that they're building. So what is build a cell? Cells are the fundamental building blocks that make up living organisms. Yet we don't know exactly how cells were formed in the first place. We also don't know what all the molecules that make up any natural cell do. So, we have a lot of genes being expressed and proteins being produced and things in the cytoplasm, organelles and structures in the cell, but we don't necessarily know what they do and how they sort of interact. I mean, we kind of have some idea, but this is something that, of course, is very hard, especially when you move from cell type to cell type or organism to organism. Finally, we can't yet put molecules together ourselves to make new synthetic cells, and that's really the thing. We haven't really been able to master kind of building a cell, and what that means is basically, building a cell from its components, not just taking a cell and modifying it, but building a cell on its own. So we've done things like taken bacterial cells and derived sort of a minimal genome, and then basically stripped out all the genes except for that minimal genome, and we've had those cells, we've been able to culture those cells successfully. We've also been able to um, do things like have a sort of a vesicle that has components in it, and what they call a minimal cell, and allow that vesicle, which is like an open space with a membrane, allow that to have some sort of metabolism and study those sorts of systems. But those aren't really cells in the sense of like a cell that you would find in your body. So these are very minimal systems that are very limited functionally. So this is what they're trying to do: is they're trying to build something that is really something that we would find in the body. We can use it for studying disease and other things like that. So they want to. The future of biotechnology is in realizing fully understood lineage agnostic organisms, beginning with a single cell. So they want to build things that are not tied to lineage or lineage trees like what we have in. We have in C. elegans is very lineage dependent. Cells have to be of a certain lineage, and that defines their function. In mammalian cells, usually there's some flexibility in terms of what a cell can be. So we can take cells and reprogram them to different fates. But by and large, their cells are usually restricted to some sort of pluripotent set of fates. We do have cells that are totipotent, but those are harder to work with in terms of building very specialized cells. So building cells is hard, but if we could better work together, then we should be able to build many cells for many purposes. So this is an open source community, basically, around this goal and trying to achieve this goal. So this is a current but brief overview of the synthetic cell field. It gives you some information about synthetic biology and what people are doing to build synthetic cells. There's this paper, this comprehensive roadmap summarizing recent progress and defining the most urgent needs of our field. This paper is from ACS Synthetic Biology. This kind of goes over, Lynn Rothschild is one of the authors on it. She's actually a pretty big name in astrobiology and in some of this sort of stuff. I attended a workshop last year on was Symbio, which was held at the University of Minnesota, and they kind of talked about a lot of this stuff. So I think I reviewed that in one of the meetings from last year. So this is kind of in this area. So this paper kind of talks about the different challenges that are sort of at the forefront of this field. The de novo construction of a living organism is a compelling vision. Despite the astonishing technologies developed to modify living cells, building a functioning cell from scratch has yet to be accomplished. So we haven't been able to build a cell from scratch, and we've just basically been able to take parts and manipulate them, but not build something from the sort of the proteins or the raw DNA or whatever, and build a cell. And so that's kind of the goal. It's worth asking whether that's achievable. And so that's maybe one of the challenges here. The pursuit of this goal alone has and will yield scientific insights affecting fields as diverse as cell biology, biotechnology, medicine, and astrobiology. Multiple approaches have aimed to create biochemical systems manifesting common characteristics of life, such as compartmentalization, metabolism, and replication, and the derived features of evolution, responsiveness to stimuli, and directed movement. So we have these sort of structural aspects of cells, compartmentalization, which means that you have organelles, you have, you have an empty sort of vesicle, you have a membrane with organelles, you have a nucleus that forms, you have, processes that happen in the cell, like DNA replication, transcription, translation, protein assembly, and so forth. And those sorts of things are all sort of the structural characteristics of the cell. You have those things in service of metabolism or replication. And then you have things like the evolution of a cell, the responsiveness to stimuli, and directed movement, which we know from a lot of the stuff that we've talked about with respect to epithelial sheets is very important in development. Significant achievements in synthesizing each of these criteria have been made individually and in limited combinations. So they review a lot of this stuff, and they identify the bottlenecks in the current research, and they produce a roadmap. So I don't think I have access to this right now, but this is something that, if you're interested in this area, would be good to kind of go over and get familiar with. So they have a lot of nice resources here. They have this video on engineering synthetic cells, which I won't play. And I think what Morgan was highlighting is they have a seminar series. So they have the seminar series here, which is I don't know if they have the sort of a playlist or anything, but they have these they have the workshops, they have this seminar series. And, there is a YouTube channel. Okay, so that's good. And they have an in silico cell working group and a build a cell chemical reaction network set of modeling tutorials for chemical reaction networks. So this in silico cell working group is working on these chemical reaction network tutorials. The build a cell workshop is, so there's one coming up in Boston, workshop number 13. And these workshops, I guess, go, back to, I don't know when. This might have been the one actually that I attended. This is maybe associated with the SynBio conference. This is, let's see if, they have, I think, a little bit of a, they don't really tell you what these are, but they have breakout rooms here, so, yeah. Okay, so they do a lot of interesting things at these workshops. They talk about different topical areas. Let's see if they have anything on those working groups that may be useful. So they have this. They have these working groups. One is Life OS, which is building an operating system for life. This is where you're basically trying to define this. How to kind of have an operating system for a cell. If we remember what an operating system does for a computer, it allows programs to run on a computer. So it would basically interface with like maybe the genome and some of the metabolic processes to sort of allow you to program things in the cell. And this is again a metaphor, so we don't know exactly what that would look like. If there's going to be like a graphical user interface for that, but that's Life OS. Then there's the in silico cell group, so they're doing these types of things. They're working on models of cellular subsystems, benchmark datasets, and Turing tests to measure the viability of models across physical scales. So basically, if you have like, you talk about maybe how you can do some sort of set of computations in a cell and, have tests that, show the viability of some sort of computational approach. And then doing this at different physical scales. So from the atomic scale to the whole cell. So, the cell ultimately is composed of a bunch of atomic processes, but you have to link that to things that are going on at the cell level. Then they have visualization methods and games and competitions to enable decentralized modeling. I think that's more along the lines of organizing the community. Interlab groups here. There's ethics and biosafety, and there's this translation group that seeks to translate these findings out to sort of the medical community, making sure that they get applied. The essential genes group is where they look for these minimal gene lists. So basically you take a list of genes and you test each gene to see if it results in a viable cell. If you knock out a gene and there's no viable cell, then that gene is essential. If you knock out that gene and the cell lives, then that's not an essential gene, and you can get rid of that. And so you end up with this minimal gene list that allows you to boot up a cell in sort of its ground state. And, it isn't like, it isn't a very robust cell. It just allows the cell to be able to be alive and maybe replicate. So then once you have a minimal gene list, you can then add genes to boot up different functions. So if you add in you have a minimal cell and you add in a pathway for some sort of metabolic enhancement, that would be, taking that minimal gene list and adding to it to produce something that can be, you can think of that as maybe a program. So this is how they're kind of approaching this. And then there are, of course, synthetic cell gene repositories where they give like the recipe for this. They have them sort of in a repository here. And so that's those are the working groups for Build-A-Cell. So that's, I think, really useful in kind of thinking about how people are approaching this and, the kinds of challenges that exist in this field. So again, this is the Build-A-Cell group. And, there's Susan and Morgan is here. So Morgan, we were just talking about Build-A-Cell. I don't know if you had anything to add with respect to that. Seems like there's a lot more than just the seminar series. Okay. Hello, Susan. I don't know if you're available to talk, but I wondered if you had any updates you wanted to give.
Sorry, I'm just taking a note on Build-A-Cell.
Okay.
So, sorry I'm late. It's a holiday here, so we're I slept in actually. Updates, I don't know. Console's still being. It's not giving me results. I watch YouTube videos and it says it works this way, but I think it's for an older console. And my help desk has decided to quit entirely. Oh. It says 404 error now. So I was like, I really need to do chat GPT today.
No.
It'll give me that one last little hit.
Yeah.
I just wish I could talk to someone who has used the program.
Yeah, it's always hard.
Yeah. Anyway, nothing much. Doing a lot of gardening instead of homework.
Yeah.
Right. Anyways.
Yeah. Right. Yeah, and Morgan said that it's really a nice seminar series, Build-A-Cell, and So go ahead.
Do you have the URL for it?
Yeah. buildacell.org. Let me put it in the chat. There you go. All right, so the last thing I wanted to talk about today was this paper on spectral decomposition of embryos. It's really interesting work. So this is actually an eLife. The way they do eLife now is they have they allow you to preprint things, and then they review them, and then eventually, if you get published, it becomes like a published paper. So it's a kind of a transparent peer review model. So I think they're at the stage of being reviewed preprint, but it hasn't been revised. So, but I think this is a really interesting paper. It kind of goes through some interesting things that you can do in embryos to look at spatial variation and some of the information that you might have. Break down what's going on in a embryo into this sort of frequency spectrum, and then decomposing that and understanding different types of morphogenesis. So this paper is Spectral Decomposition Unlocks Ascidian Morphogenesis. And Ascidians are otherwise known as sea squirts. And aside from C. elegans, another organism that exhibits a lot of similar traits, especially in early morphogenesis or early embryogenesis, is the sea squirt or the Ascidian. And fortunately, we have a lot of data on this early stage of Ascidian morphogenesis. And it's something that we've explored in papers in the group here. We had a paper from 2016 where we compared Ascidian morphogenesis and C. elegans morphogenesis. So there's this, like C. elegans, you have these cells that start with a single cell, and they differentiate, or they divide into these sort of clonal cell types. There's a very specific nomenclature for the cells, and it's predictable, highly predictable from one embryo to the next. So you can look at this early morphogenesis that results in this large ball of cells. Eventually, though, Ascidians are going to, the Ascidian embryo is going to differentiate into this long tubular tadpole that actually doesn't look anything like its early morphogenesis. But the part of morphogenesis that's most interesting, at least from the standpoint of this spectral decomposition technique, is this very early stage. So keep that in mind when I show you the images, because it's, it just gives a little bit more context. So this group here, Patrick Lemaire and some of these Edmund Rowe, they work on this Ascidian morphogenesis. And this community, like the C. elegans community, they have a lot of good open data sets for what they're doing. So that's good too. So the abstract reads, describing morphogenesis generally consists in aggregating the multiple high-resolution spatiotemporal processes involved in reproducible low-dimensional morphological processes consistent across individuals of the same species or group. So they're basically taking different spatiotemporal processes, and then you're trying to reproduce some sort of statement about morphogenesis. And, you can't really maybe try to do it across species, but usually it's species specific. That's what they're getting at this sentence. In order to achieve this goal, biologists often have to submit movies issued from live imaging of developing embryos. So this is kind of the standard that people use. We take microscopy movies a lot, and those are usually the most informative ways to look at morphogenesis. We certainly do that with C. elegans, and what we do when we want to do machine learning is we take those movies, take it frame by frame, and train the model on those movements or whatever that we're looking at. So we're looking at division events, we're looking at movements within the embryo. We can take that from this time series of images and describe the movement from frame to frame. So how much is something displaced from frame to frame? That gives us a good indicator of movement or of dynamics. So what they're saying is that that's sort of the standard in the field. These approaches, however, present noticeable drawbacks, as they can be time-consuming, hence unfit for scale, and often lack standardization and a firm foundation. So these are all challenges we've seen in the C. elegans datasets. In this work, we leverage the power of a continuum mechanics approach and flexibility of spectral decompositions to propose a standardized framework for automatic detection and timing of morphological processes. So they're looking at a continuum mechanics approach, which means taking the embryo and the changes in the embryo and putting it into this Lagrangian model, which is a continuum mechanics model. So you write a Lagrangian out, and the Lagrangian describes movement in space, and then you can use that type of a model to derive, you can derive things like we talked about last week. You can derive calculus, and you can derive other types of continuous mathematics to analyze these. So in this case, they're looking at spectral decompositions and how to take that time series or those dynamics and describe it as like a spectral, like a power spectrum or something, and then decompose the information there to get, to say something about morphogenesis. First, we quantify whole embryo scale shape changes in developing Ascidian embryos by statistically estimating the strain rate tensor field. So this is something that we're interested in, and it's of its time evolving surface without the requirement of cellular segmentation. So a lot of times in the C. elegans stuff we're doing with the machine learning is we're taking apart these movies, we're kind of using segmentation algorithms to find those features, and we link those features across the frames in time. We maybe count the centroid and we say, what's the displacement of a centroid from frame to frame? And then we get this, so we use these cellular segmentation techniques as a guide to how to track things in the movie, how to track things over time. But what they're saying in their approach is that you can use a strain rate tensor field to build this time evolving surface that gives you similar information or maybe different information. And so this is something that you can use as well. It's more mathematically complex, but it gives you it's a good approach for looking at these things. We then apply to this data spectral decomposition in space using spherical harmonics and time and using wavelet transforms. So they use a combination of spherical harmonics and wavelet transforms to get the information out. So wavelet transforms are, where you take a time series and you decompose it into its high frequency and low frequency components. You do this with different types of mathematical transforms, and you do this at different scales. So you can take like a very long time series and decompose it using these high and low frequency filters that sort of emphasize the high frequency and the low frequency components of the time series, and it gives you sort of this characterization of maybe in a very small time series. It summarizes everything, the basic changes. So for high frequency events, it emphasizes those high frequency events. It shows if there's key high frequency events occurring in the time series, it'll kind of identify the part of the time series where that's happening, and the same with the low frequency components. So that's a nice approach, but it's very hard to implement, and that's kind of where they're going to go with this. You have to use kind of a system like Ascidians or C. elegans to do this. Otherwise, I think it would probably not work very well. These transformations result in the identification of principal dynamical modes of Ascidian embryogenesis and the automatic unveiling of its blueprint in the form of spectrograms that tell the story of Ascidian embryos. So that's what they're doing. They're building these spectrograms from these principal dynamical modes. So this is a lot of sort of dynamical systems analysis and looking at the dynamics of the embryos instead of using the kind of features that we're used to using. The feature-based approach is easier to implement, but it doesn't give us the same information. So this is they talk about this approach. Embryogenesis can be described as a series of stages delineated in space and time by the identified landmarks, and that's sort of the standard in the field. And so, this is sort of congruent with our feature-based approach, because you're basically finding these landmarks in the embryos morphology. You're using those landmarks then to connect between the landmarks and look at how the embryo changes. And that's something that can give you some information, but not the full story. So, they go through some of the standardized methods, the sort of contemporary methods, and then they kind of talk about some of the challenges of that, because a lot of the processes in morphogenesis are inherently multi-scale, and this is true both in spatial and temporal domains, which these multi-scale processes also interact and overlap. And as in the case of several species, Ascidian gastrulation is a playground featuring important displays of cellular divisions and tissue mechanics, most notably endoderm and vagination. So there are these processes that occur, these collective processes that are collective behavior across cells, that we're interested in, these kind of large-scale movements that are hard to capture using this sort of landmark or feature-based approach. So they're basically trying to differentiate between local and global sort of behaviors and make distinctions between them. Then they talk up spectral decomposition, and these are the references here: Romeo et al. 21 and Del Mazo et al. 21. Spectral decomposition, where a signal is broken down into its canonical components, is well suited to the study of systems that exhibit multimodal behaviors. The benefits are at least twofold. The first is that individual constituents may represent distinct dynamical processes, thereby enabling the decoupling of physical processes entangled in the data. And two, only a handful of components may significantly contribute to the original function, resulting in a compressed lower dimensional representation that capture the main features of the studied process. So you have this thing where you have parts, and they can contribute to the whole on their own. But you also have these situations where you have a handful of components that contribute more than other components to a global process. So you can actually extract those, the effects of those components, or you can look at sort of this, the individual components kind of additively. So there are a lot of things you can do here to sort of get information out of the embryo. So without further ado, I'm going to go down here and so they use these Lagrangian markers on the surface of the embryo. So they build this Lagrangian model of the embryo. And this is a continuum description. So it allows us to have this strain rate field and characterize the embryo as a strain rate field. So things are happening in this field across different cells, and you're getting this model that is continuous rather than discrete. So I want to go down to the images. This is the Ascidian embryo, so you can see it's this clump of cells, and this clump of cells actually has many-fold symmetry. So you have, I think it's four-fold symmetry, where you have each quarter of this sphere has sort of the same cells, the same structure. The divisions happen they're binary divisions, but they end up giving you this four-fold structure. So this is an interesting feature of this embryo. So just to keep that in mind. So the first stage here is the level set scheme for evolving surface generation. They build the strain field here. They have a dynamic. They model the dynamics with this equation here, and this is the math. Then they have the raw data where you get a resulting mesh. So they're trying to render an embryo surface from the raw data. You have the scheme for evolving surface generation. So now once you have the data and you have the surface generation model, then you can validate this by tracking cellular junctions. So this is something that is relevant to what Susan's doing. You have these cellular junctions in red, where you have the sort of where the cells meet together. And so remember, you have the raw data from the cells. You have this mesh that you build, so you're not just relying on features and their centroids. And then you have this model of sort of a strain field. So things are being transmitted across the different parts of the embryo. Not necessarily the centroids, but things like at cell junctions. And so here they have the cell junctions from raw data. They plot this on the mesh, so they're able to measure using these strain fields the cell junctions, and they're able to apply this to the mesh. Then you have these simulated cell junctions, so you can both take things from raw data and simulate cell junctions, and then you can find the Lagrangian error between these two components. So you have the cell junctions on raw data, the simulated cell junctions, and then you have this Lagrangian error. So the Lagrangian builds you this time series where you have the cell junctions and the simulated cell junctions, and they diverge. And so there's this error that you get, and this shows the error between the simulation and the observation. So you see the two components, the red component here and the green component here in the equation. And so that's what it looks like. And then you can do some interesting things. So you can plot out relative displacement between cellular junctions and their markers over time. So you have the time component here. This is a time series where you see the average displacement. You have the cell count that goes up. So this is one of these graphs where the average displacement is on the left and the number of cells is on the right. So as this number of cells increases in this model, and it goes from 0 to about 256 cells roughly. And this is just in time, this is from 4 to 5.75. So it's like a fraction of this time series. And so you get capture most of the division events in this time series, but you can see the average relative displacement fluctuates quite a bit during this process. And in fact, it looks like there's a sort of a peak in the average relative displacement right before there's a burst of division activity. So you can see that, well, at least for here, you get this burst of division activity. Whereas here you don't see that as well, but you see it here and here. So you get these interesting, signatures that are maybe, something to follow up on later. So you get some really interesting findings out of these kind of models. So these are what they call level set inspired Lagrangian markers. And that's what they're doing up at the top. And then they're applying a mesh. And then they're able to both observe things from the data and simulate these things. And then they're able to find the Lagrangian error, which gives us these kind of graphs. The second thing here is where they talk about the strain rate field, and it describes Ascidian morphogenesis. A correct parameterization of the velocity field at every position requires 3 coordinates. And this is in Vx, you have Vxx, Vyx, and Vzx. So you have this, basically this 3-dimensional model. The strain field is derived as the symmetric part of the discrete gradient of the velocity field, as described in Banchalia 2018. Intuitively, the strain rate field, the strain rate evaluated on a given mesh vertex measures how the velocity vector varies in the neighborhood at this point. So this is the point here, and this is, I guess, how the velocity vector varies depending on the symmetry and what's going on locally in the embryo. So this is, allows us to look at these local variations in the embryo. And then they talk about the mathematical construction of these different axes. The diagonal elements of the matrix capture the linear strain rate in the XYZ axis, depicting the change in length per unit length. The 9 diagonal elements stand for the shear strain rates in the XY, XZ, and YZ directions. Because D is symmetric up in this equation here, there exists an alternative representation which holds stronger local geometric meaning. So you have this local geometric aspect that's really interesting. And so this is another image here where you have the strain rate tensor field, or the tensor strain rate field, and that's here for these different components. So you can see that. This is an example of this one here. So this is an example of the strain rate tensor field across the entire embryo. Then you have a scalar strain rate field, which is this here. So these are 2 different measurements, the tensor strain rate field given by this equation and the scalar strain rate field given by this equation. And you can see that it gives you these different pieces of information. And this is all, this is sort of a phase portrait of the embryo, so you can get, different information from this for different points in time. So the strain rate tensor field measures the rate at which morphological changes occur in the embryo as a function of time. The strain rate tensor field is locally represented as a 3x3 symmetric matrix and is completely determined by its eigenvector fields. And so this is a heat map in A, and then in B we have a heat map of the scalar strain rate field. So these are heat maps, but you can use them to determine dynamical modes as you wish. And so yeah, they give more information about their methods here, and they talk about spherical harmonics, revealing the main modes of Ascidian morphogenesis. So in order to capture the relevant features of the strain rate field in the spatial domain, they can conduct a spectral analysis of the scalar strain rate field. So this is, down here we have sort of this decomposition. So I think these are the different, let's see. Yeah, these are the different, spherical harmonics here. So you have these different modes where you see these different sets of values. So you kind of average it out, I think, basically down to L equals 0, L equals 2, and you can get you can see what they look like at different scales. You're decomposing the spherical harmonics. And then you have this dominant modes of morphogenesis where you have the variance ratio as a function of percent and the number of cells. So we have Y00 and Y10, and we can see what those behave like given the number of division events, the time. And then we have this time series of F00 and F10, which they're kind of consistent, but their value here in this y-axis is a little bit different. So it's interesting the kind of information you can get out of this. So I think in summary, we can look at the wavelet transform. So this is figure 4 of the wavelet transform, where we have F00, which is this for gastrulation and neurulation period. So these are different periods of embryogenesis where you get these different types of folding, and so neurulation is the formation of the neural tube. Gastrulation is a type of invagination where you're getting a change in the shape of the embryo. So this is the wavelet transform, and again, you're doing this wavelet transform, and you're taking these components and you're plotting them out in this kind of map where you can actually identify these different dynamical modes. So you get the duration in hours here. So you go from the top, which is the 1 cell stage, down to 4 hours, and then you get this these HPF values, which are from 4 to 8.4. So you get this invagination here in this region, you get zippering, you get synchronized divisions up here because you get this at early stages of cell division, you get these synchronized divisions that you can actually clearly see here, and then you get this change in shape. So you get these changes in mass, which are symmetrical, and then you get these asymmetries like invagination and zippering that occur, and you can see the modes here. The same thing here where you get these different movements, you get synchronized divisions up at the top, up at the beginning of embryogenesis, and then you get things like endoderm apical basal shortening and mesoderm cell movements. And you can see those as sort of these phases. You have the synchronized divisions again, and you get endoderm plus mesoderm morphogenesis down here, so you start to get differentiation as a function, as you go longer into the process. And then this is for invagination period here for FT20, which is, these are, I think, decompositions of the spectral decompositions, and then putting that into the wavelet transform. So you're doing 2 types of decompositions. You're doing this type of spherical harmonic decomposition, and you're doing the wavelet transform, that which also decomposes the time series. And so you end up with this endoderm apical constriction, endoderm apical basal shortening, which you can identify in the same manner that you can identify these other kinds of global processes. And so this is a really interesting paper, I think, kind of describing a different way to do this kind of analysis. Like I said, it's a pretty mathematically involved way, but I think, if you think about the physics of the embryo and thinking about like modeling things from single cells to these large-scale movements and other things, it's a valuable approach.
Okay. Do they have pictures of the inside of the embryo?
I think they're just modeling the outside. I mean, the inside is, I think, basically symmetrical. I'm not sure how they're, when you look at these data sets, they kind of describe the cells as these, at the stage that we're looking at, as these fourfold symmetric structures. So each quarter of the embryo has certain cells in place, and then they have, there are 4 of those. And the outside is the thing that's going to change. So as you get these movements, it's the outside of the cell mass that changes, and so that's what they're modeling with their fields.
They need to look at the inside too, and the technique for doing that has been around for 5 years.
What is that?
Optical coherence tomography.
Oh, yeah.
Yeah, and as the inside becomes fluid filled, that affects the cells on the outside. It's a very important part of this.
Yeah.
So they're analyzing something, but they haven't looked at the density of the embryo or the pressure on the inside of the embryo. Just think it would be good to put with that data set.
Yeah, it would be. And they don't talk about the sort of the material aspects too much. They just kind of talk about the...
I'm glad they have cell junctions. Those are important.
Yeah. I thought you'd like that, yeah.
Yeah, I like that. But there's still it's sort of like, sing, oops, they missed something.
Yeah.
Oh, well.
Yeah. Okay. Well, I think that's all for today. Do we have any other comments or questions? Okay. Well,
I'll look at the recording because I missed the first part.
Oh, yeah. Okay. Well, thanks for attending, and have a good week. Thank you. Bye.
Yeah, thanks.
Thanks.
Bye.